Hello students, I'm C. Pavani, Assistant Professor at BVRIT Narsapur from Pharmaceutical Engineering Department. In today's video, we will learn about omega oxidation of fatty acids. Coming to omega oxidation of fatty acids break down by various mechanisms. Beta oxidation is we have already learned in the previous video. Now we will learn about omega oxidation. So mostly beta oxidation is the first and primary importance for the oxidation of fatty acids. If there is any failure in the beta oxidation mechanism, then the fatty acids will convert for the omega oxidation process. So we can say this as alternative pathway for beta oxidation process. And generally beta oxidation occurs in mitochondria of the cell. And this omega oxidation occurs in smooth endoplasmic reticulum of the cells present in kidney and liver. As you know, a cell contains cell membrane and mitochondria as well. So you can see beta oxidation in mitochondria, and around the nucleus you have endoplasmic reticulum. There are two types of endoplasmic reticulum, that is rough endoplasmic reticulum which contain ribosomes and smooth endoplasmic reticulum which do not contain ribosomes. Our omega oxidation of fatty acids occur in smooth endoplasmic reticulum. Remember these areas, whereas beta oxidation occurs in mitochondria and omega oxidation occurs in smooth endoplasmic reticulum. And in this process of omega oxidation, you can see as we know, fatty acid contains more than 10 carbon atoms in its structure, and fatty acid contains carboxylic group at one end, and the carbon which is present adjacent to the carboxylic group, you will call it as alpha carbon atom, and adjacent to alpha carbon atom the carbon is called as beta carbon atom. Oxidation process at this place is called as beta oxidation process. Omega carbon is nothing but the last carbon of the fatty acid. Whatever the oxidation occurs at the last carbon of fatty acid is called as omega oxidation. And the omega, so as you can see, methyl group will be present at the last carbon atom. A fatty acid which contains methyl group at the end of the chain is considered for the omega oxidation of fatty acid. There are three steps involved in this omega oxidation. First step is hydroxylation. So in the first step of this omega oxidation, the end methyl group is hydroxylated in the presence of oxygen, molecular oxygen, as well as NADPH. This methyl group is converted into hydroxyl group by the addition of oxygen at the end. And this occurs in the presence of cytochrome P450 monooxygenase enzyme, which is present in the endoplasmic reticulum of the cell. And coming to the second step, it is oxidation. Oxidation here, the hydroxylation, after the hydroxylation step, the hydroxy fatty acid, this is hydroxy fatty acid, that is fatty omega hydroxy acid, gets converted into aldo acid. By this, you can see this is aldehyde group. This conversion takes place in the presence of alcohol dehydrogenase and also in the presence of NAD+. NAD is nothing but nicotinamide adenine dinucleotide, and this is an energy source which helps in the process of this conversion. And for this step, we require enzyme, that is hydroxy acyl coenzyme A dehydrogenase. So this enzyme helps in the catalysis of this reaction. Third step is cleavage. Cleavage is nothing but breakdown of fatty acid chain, and this cleavage step requires thiolase enzyme for the progress of the cleavage, and this is also a last step of beta oxidation pathway, and you can see two fragments after this cleavage step. Here you can see fatty omega aldo acid in the presence of aldehyde dehydrogenase, as well as in the presence of nicotinamide adenine dihydrogenase. It gets converted into fatty dicarboxylic acid as well as acetyl coenzyme A. As we discussed, after the cleavage, you can see two fragments of one single fatty acid. So the two fragments are fatty dicarboxylic acid, where you can see carboxylic acids at both the ends, and you can see acetyl coenzyme A as second product. So these are the further metabolism occurs, where you will have end products as two dicarboxylic group containing fatty acid, as well as you have acetyl coenzyme A as end product. The acetyl coenzyme A produced in this is utilized for the further energy, like it gets converted into fatty acids. It can be utilized in Krebs cycle, as well as it can be utilized in further biological mechanisms. And the dicarboxylic acid generated after cleavage of this fatty acid is further used in the metabolism of other cellular requirements, and it is also used as a precursor for many amino acid synthesis. And alternatively, this dicarboxylic acid can also be converted into metabolites through various enzymatic reactions. So let us see the overall process. So in the first step, we call it as hydroxylation step, where fatty acid gets converted into fatty omega hydroxy acid in the presence of cytochrome P450 enzyme. And in the second step, fatty omega hydroxy acid gets converted into fatty omega aldo acid, where this happens in the presence of alcohol dehydrogenase. And in the third step, fatty omega aldo acid gets converted into fatty dicarboxylic acid plus acetyl coenzyme A, and this happens in the presence of thiolase because it's a cleavage step. The biological significance of this omega oxidation steps is, it is a minor pathway. This happens only when the beta oxidation process is defective, and some studies have shown omega oxidation can also form glucose from succinyl coenzyme A during starvation and diabetes condition, and it is important pathway for the production of pheromones in honeybees as well as biopolyesters in higher plants. Thank you.
Like, share and subscribe. Hit the bell icon for more updates.